Today's presentation will focus on cancer DNA testing. We can divide cancer DNA testing broadly into two types of categories: those that are impacting cancer patients directly, or patients who have had cancer already, as well as family members with history of cancer. And if we focus on the latter group, your predominant test available to you is one that will assess you for your genetic predisposition to cancer. So you want to find out whether you have the risk of developing cancer based on the genetics you might have inherited from your parents. And cancer predisposition DNA testing will be the focus of this presentation. But I also wanted to bring up the repertoire of tests available to cancer patients as well, just to show you how complex this field can be. So to give you some examples, the most basic test that would be available to cancer patients, and also the cheapest, would be a test what we refer to as pharmacogenetics test. And this type of test simply assesses the patient for how they react to medications, including cancer-related medications. So this type of test should seriously be considered by any newly diagnosed patients, so they can determine whether the medications that is going to be prescribed to them is actually safe or not. Next test is also predisposition testing, and in this case it's not just to find out whether other family members related to you might be at the same risk as you, but this type of test can also inform the cancer patient on potential treatment options. Next in line is the most complicated and sophisticated type of test, and that is where the DNA of the cancer itself is being sequenced to determine what kind of genetic mutations actually contributed to the development of that cancer. This is very expensive test, runs in multiples of thousands of dollars, and the reason why is because it requires extra power to be able to fish out those cancer genetic mutations out of a sea of normal cells. Then there's also another type of category of tests that is referred to as liquid biopsy. This is still quite an emerging field in cancer DNA testing, and basically what we would be looking at here is the cancer DNA that is directly floating in patients' blood, as well as potentially cancer cells that are floating in patients' blood. And all of these tests will be performed for singular purpose of determining how the cancer patient should be treated specifically for the type of cancer they have based on the specific DNA fingerprint of that cancer. So we are talking about here selecting personalized medication. But before we focus on the DNA testing for cancer predisposition, I wanted to give you a bit more background on these type of different cancer mutations so the idea can become more clear. So the DNA that you inherited from your parents is the blueprint that determine how you developed and also how you are run from day to day. And half of that information presented here as chromosomes, which are structures that tightly package DNA, you obtain from your mom, and the other half you obtain from your dad. We have 23 pairs of these chromosomes, 22 of which are identical, And the last pair is referred to as sex chromosomes. And you always obtain X chromosome from your mom, and from your dad you can obtain either X chromosome or Y chromosome, and which one you will receive from your dad will determine the gender of the baby. But along with that DNA, you also inherit genetic mutations. We literally receive thousands of these. Some, of course, you get from your mom, some of which you get from your dad. And there was nothing you could have ever done about this. It's a natural process until now, and I will touch on that later on. These days, you can take advantage of available technologies and decode all of this information to learn what these mutations are through the process of DNA sequencing. What the public is most familiar with is DNA sequencing to obtain personal traits or ancestral information. However, this is not very medically useful information. You can also obtain information, though, that can be clinically useful. So I already mentioned pharmacogenetics, which is learning how you can be using medications, specifically for your own biology. Another type of information is what is referred to as carrier status. So these are the type of mutations that you might have that will not impact you, but if you and your partner combine same mutations, then you run the risk of developing a disease in your baby. So this type of information is very valuable for reproductive planning. And finally, you can also learn how these type of genetic mutations might predispose you to different genetic conditions or diseases. And that also will include cancer. But along the way, throughout your entire lifespan, you will also pick up brand new mutations, and they will show up spontaneously. They can be caused by your environmental exposure. So one example would be cigarette smoking or UV light, although majority of these will occur naturally due to mistakes during cell replications. So there's not much we can do about that. And eventually, if enough of these mutations, new mutations, show up, and depending on where they show up in your DNA, they can cause cancer. At that point, as I already previously mentioned, this type of information can also be decoded through DNA sequencing for the primary purpose of determining how a specific cancer should be treated. And indeed, this information would be specific to a given cancer type. Just to give you an idea of how many genes are involved, by one type of source, we already know there's at least 280 genes that are involved in cancer predisposition. So this is the type of information you're already born with. And we know of at least 600 different genes that, if mutated, could be contributing to the development of cancer. So there's many different ways of how cancer could be developed. So the take-home message here is that inherited mutations predispose you to cancer, whereas the acquired mutations usually, almost always, will be the required trigger for cancer to actually develop. So now let us go back to the type of DNA tests that were mentioned in the very beginning, but now cancer patients, we will split into two categories, those that are referred to as currently diagnosed cancer patients, as well as patients who have survived their cancer and have had their cancer successfully treated. And the final group, of course, is, as we mentioned, are people with cancer family history. So previously we combined the diagnosed and surviving cancer patients together. So for diagnosed cancer patients, just to review once again, the type of tests, DNA test tests that are available to them includes pharmacogenetics, tumor biopsy, DNA sequencing, as well as liquid biopsy. And these types of tests are there to determine how a newly diagnosed cancer patient should be treated. For surviving patients, they, in theory, have an option of liquid biopsy. And the purpose of this would be to monitor whether cancer is coming back. This is fairly tricky, so how effective this process is still a matter of debate. And the reason why is because you have to know what kind of mutations you're actually looking for. So this would only be available to patients who survived cancer who already knew what mutations developed their original cancer in the first place. Otherwise, you'd be randomly trying to find something, and as I already pointed out, there's literally hundreds of genes that could be affected in cancer development. But theoretically, this option is indeed available. And then, as I already mentioned, with family history, any member of such family has an option of determining whether they have a cancer predisposition, so this type of testing is done for screening purposes. Now, this last option that I'm talking about is obviously also available to diagnosed cancer patients as well as cancer survivors. And as I already previously mentioned, specifically for diagnosed patients, looking at genetic predisposition to cancer can also inform about treatment. And usually when diagnosed cancer patients undergo tumor biopsy DNA sequencing, that information is done in conjunction of DNA sequencing of the genome or genetic information that they were born with, just to gain that extra insight as well. So this summarizes broadly what kind of DNA tests are available for cancer, and now we will focus specifically on predisposition. Now let's go through the type of DNA tests that are available to you for screening for cancer genetic predisposition. In order to obtain this test, you would have to provide either blood or saliva, because we are looking at inherited DNA, and both blood or saliva will provide plenty of that DNA to be screened. And in broad categories, the simplest type of test is a test that looks for specific selected mutations. This type of test should only be done for people in families where a member of a family has already been identified that they have a genetic predisposition, and that specific mutation is already known and other family members are screened for it. It would be inappropriate to use this test for discovery purposes because it can lead to very large proportion of false negatives, and I will explain that later. The most common type of test available to people is looking at specific genes of interest that are pre-selected based on well-confirmed knowledge of which genes contribute to cancer predisposition. These are referred to as gene panels and are most commonly used in medicine. Next option would be to look at your entire genome, which is looking at all of your DNA that you were born with. And finally, it would be any combination of the above tests with additional extra tests. And I'm showing you here example of RNA testing or polygenic risk score. And I will explain that in detail in just a moment. As you go from top to bottom, the cost of those tests increases, as well as the amount of medical information that is obtained increases. The purpose of these tests is for a person to identify whether they have genetic predisposition, and if they find out that they do, they will want to then monitor themselves more frequently for earliest development of cancer. But this information can also be very valuable for reproductive purposes, so family planning, because if you know you have a genetic predisposition, you have to weigh the risk of passing on that information to your child. However, it is now feasible to eliminate that risk. This process is referred to as pre-implantation genetic testing, and that's where fertilized eggs can be screened whether the cancer predisposition mutations have been passed on or not, and only fertilized eggs that do not have mutations could be then selected for implantation and future pregnancy. Let's discuss the limitations of DNA tests that look for specific genetic mutations. The most famous example of that is the direct-to-consumer 23andMe test, which means that anyone can purchase this test online. And this test does indeed look for few specific mutations that could predispose an individual to cancer, specific type of cancers too. But what a customer has to understand is that you are only going to be screened for very limited number of mutations, literally just a few of them within specified genes. But the reality is that within those same genes, there's literally hundreds of different possibilities of how those genes could be mutated and leading to cancer predisposition. And therefore, a customer has to know that if they obtain a negative result, meaning they did not obtain results that tells them there's a predisposition, they're not out of the woods because a mutation could be lying somewhere else within that gene. That's what I meant by previously by mentioning that there is large risk of false negative results. And for this reason, it is always recommended that any cancer predisposition tests should be ordered by a doctor so that there is a proper medical oversight for patients, and so that patients could be properly explained of what the limitations of any given tests are, so that the patient does not run the risk of being misinformed. 23andMe test is highly accurate test, but the customer has to be aware of what the test actually looks for and its limitations. Now let's get the background for the value of supplementary RNA testing. So here we see a stretch of DNA, and along the DNA there are segments that are referred to as genes. And genes are copied from the double helix DNA into a shorter single strand that is nearly identical to DNA. It's slightly different chemically, and this is what is referred to as RNA. And it is only the RNA that actually acts as the blueprint to develop proteins, which basically are tiny little molecular robots that do all the work within your cells. But this is the simplified version. The reality, of course, is a little bit more complex than that. In reality, genes are not a contiguous blocks of information. Rather, they're split apart into a number of tiny little segments that then have to be spliced together into a single RNA message. Why does this exist? It's because this allows a single gene to be manipulated and spliced in different fashions in order to produce different products. So you can combine all the fragments together and that will produce one type of specific protein, or you can select different components of that same gene and it will produce something else, and so on. Some of which we might not even know what these proteins still do. But this basically enhances the complexity of what we can define from a single gene. You can then appreciate the fact that in order to produce a functional RNA blueprint that will then eventually produce the proteins that need to do all the work within your cells, there has to be a proper mechanism that will splice all this information together. But imagine that you have a mutation within your DNA that is not even within a gene that is involved in some important function that if this gene is disrupted, you might run the risk of cancer development. This mutation in the DNA could be somewhere outside the gene, but yet still affect proper splicing together of RNA. And if that RNA is not spliced together, you still end up with dysfunctional protein product. But you wouldn't know that from looking at the DNA directly. You would actually only learn that by being able to monitor the RNA itself. And this is how looking at RNA can enhance our understanding of genetic cancer predisposition, especially since such mutations are frequent in cancer development. So being able to screen your RNA as well can dramatically increase our understanding of genetic cancer predisposition. Finally, we are going to discuss what is probably the most abstract component to explain of this presentation, which is the polygenic risk score. So far we've been talking about cancer predisposing DNA mutations that if a person inherits such mutations, it increases their risk of development of cancer throughout life. And the reason why is because such mutations affect proteins that are involved in very important cell functions, such as regulation of cell cycle or how a cell divides, and DNA repair. And what typically happens is within each and every single cell in our body, our genetic code, our DNA, is broken or mutated literally thousands of times each and every single day. And that information has to be fixed, especially prior to cell being divided into two. So you can appreciate that if these mechanisms are affected and not working properly, how that can increase your risk of developing cancer. Now that risk can be divided into a couple categories. Number one is a category defined as high penetrance. What that means is that such genetic mutations confer very high likelihood of developing cancer if you inherited them. And the most famous example of that are the BRCA1 and BRCA2 genes. So you can see here an example of a lifetime risk for a woman if she has mutations within those genes, broadly, on average. So you can see how high this risk is, which would be in comparison to average population, which is much, much lower than this. Some of the individual mutations can confer even much higher risk than this. What you're seeing here is just an average. And these numbers can differ between different scientific publications. This all depends on what type of population is actually being tested, but this gives you a good view of what high penetrance mutations mean. But majority of genetic mutations that confer cancer risk are actually what is referred to as low penetrance, meaning that such mutation only marginally increases your cancer risk. Sometimes it could be even less than 1%. You still would love to be able to screen for all of this in order to understand how to prevent cancer development. Now, the high penetrance mutations, those are typically screened with gene panels. Those are the type of genes that are involved in gene panels that are used medically all over the world right now. Low penetrants are much more difficult to understand because it's not certain exactly how to potentially add it all up. However, certain organizations, such as private companies or research institutes, have screened so many cancer patients and studied them over a span of time that they were able to understand how such low penetrance mutations could be added up together in order to confer and properly understand cancer development risk score. And this is what is referred to as polygenic risk score. Polygenic meaning coming from many genes. And just like addition of RNA testing, addition of polygenic risk score can dramatically increase a patient's understanding of what their cancer development risk score is if such test is available to you. So hopefully this explains how polygenic risk scores work and why they are valuable as well, besides the typical DNA testing that is available to us, such as gene panels or from genome sequencing. So this summarizes what are the type of DNA tests available for cancer predisposition, and hopefully this will be valuable information for you if you choose to decide on what type of test you need. If you need assistance in being able to help you with test selection, especially from highly reputable companies, you can contact Merogenomics and we'll be more than happy to assist you and explain this information further to you. And hopefully we will see you again at another video. And if you liked what you see, please give us a like and see you next time.